Hey guys, Adam here at Society for Neuroscience 2012. Going to give you a day 4 update. It's actually the morning of day 5, but I just didn't get to it until now with this. So we talked yesterday a little bit about schemas, and today we're going to be talking, I guess, a little bit more about schemas. We're going to be talking specifically, though, about the role of sleep in memory consolidation. And so in the schema session that happened on day 3, let's see here. There was an interesting talk by Lewis about statistical learning and schemas in sleep. And so one particular study that she covered was looking at the integration of new information during sleep. And so integrating new meanings into existing knowledge, that was kind of the question. How do you do this? How do you integrate new information into existing knowledge? And so you can think that some schemas are going to be really sparse, that is to say some schemas are going to not have a lot of information associated with them, and some schemas are going to be really dense, having a lot of information associated with them, and that could make a difference, possibly, in how new information is integrated into them. So just to make this a little bit more concrete first, we can think about maybe the category B is a fairly sparse representation. Most people don't have a lot of associative information with the word B. But the word chicken, on the other hand, is probably fairly dense. You probably run into information about chickens on a more regular basis than you do information about bees. And so in this particular task, participants were asked to encode new information into a sparse category like bee, or into a dense category like chicken. And so an example would be participants would be given a sentence such as, a waba is a type of bee with a pleasant sting. And so then the participants would be allowed to sleep in the lab, and they would have EEG, that is electroencephalogram, put on them in the lab so that they could have brain recording or electrical brain recording during sleep, and look at how that would correlate with then later tests of conceptual integration learning. That is to say, how well did the participants learn what a WABA is in relation to the category B. And similar sentences could be made for dense categories like chicken. And so what they were specifically looking for in this study were these sharp wave ripples, and I'm just putting these up on screen right now. So this is electroencephalogram recording. That is, they put little electrodes all over the person's scalp, and then they can record these. They can record electrical activation that's related to directly related to neural activation in the brain. And so they were specifically looking for these sharp wave ripples that occur during slow wave sleep. And you can see here these sharp wave ripples are fairly easy to identify. They're just these areas of sudden bursts of activation, and these last about 200 to 300 milliseconds, just to give you an idea of how long these are going on. And so it's possible that these sharp wave ripples or spindles, they can also be referred to as, have something to do with memory consolidation and learning. And so they were looking to see, is there some sort of a relationship between whether participants had been learning dense information about dense categories or information about sparse categories and the number of sharp wave ripples. And so the results of this study showed that sharp wave recordings during sleep, there were more sharp wave recordings for there were more sharp wave recordings for sparse items than for dense items. So that might indicate that maybe these sparse items require a little bit more effort to encode, something like that. And additionally, they found that there was a strong correlation between the number of sharp wave recordings during sleep and memory performance, indicating that there seems to be some sort of a relationship here between these sharp wave ripples and memory consolidation. So that was kind of where the story was left off in this talk by Lewis on statistical learning and schemas in sleep. And then there was another talk, and now this was a talk yesterday. And let me just flip to my notes on this talk. And so this was a talk by Nikos Logothetis, and I'm not sure if I'm saying his name right, but hopefully. And the title of his talk was Large-Scale Brain Networks, Principles and Emerging Methodologies. And so he reviewed some very complicated methodologies, not all of which I think I fully understood. But one particular methodology that he went over, which I thought was very interesting, was neural event-triggered fMRI. And so what he had done was he had depth electrodes implanted in the hippocampus of his of monkeys. And so depth electrodes are basically electrodes just like we were talking about little electrodes on the scalp, except now these electrodes have actually been placed down inside the brain in order to allow for recording right at specific sites within the brain. And so he has these depth electrodes placed in the hippocampus, so he can see activation happening in the hippocampus at the level of single neurons even. And it turns out that there are these sharp wave ripples occurring in the hippocampus, and this wasn't something that he had discovered. This was something that was known. But what he wanted to look at was to see what sort of activation throughout the brain happens as a result of these sharp wave ripples. And so I'm going to put up a figure here. This is the Fmri activation. This is just kind of a cartooned image almost of a figure that he had in his talk. And so basically he saw that different areas of the brain, so each one of these bars represents a different area of the brain, with the black bars representing positive Fmri activation and the red bars representing negative Fmri, or that is bold activation. And you can see here that, basically the take-home message from this figure is that lots of different areas are showing random different amounts of activation. And so he had this plotted, and he wasn't really exactly sure what to make of it until he didn't really know what to make of this figure until he reorganized it based upon hierarchical stages of brain development. And he saw that basically what was going on was every time you had one of these sharp wave ripples in the hippocampus, there was telencephalon activation, with the exception of area V1, and there was diencephalon and mesencephalon deactivation. That is to say, basically the cortex was getting all this activation, but a lot of subcortical and kind of brainstem areas were getting deactivated. And so when you think about this kind of makes sense. It's kind of giving this preferential hippocampal cortical connection, so that and if you think about theories of memory consolidation, this makes sense in that theories of memory consolidation state that memories kind of move from being hippocampally dependent to cortically dependent, and the hippocampus is kind of driving that with this activation of disparate cortical areas to form extra connectivity between these cortical areas. And so it makes sense that the hippocampus would need this preferential connective state between it and the cortex. And in addition, he saw that there was specific deactivation in the locus coeruleus and raphe nuclei. And so this was also important because these two areas are responsible, partially at least, for sending people into REM sleep, rapid eye movement sleep. And that's important because during rapid eye movement sleep, there's a great deal of cortical activation. So a lot of the cortical activation that he was seeing during this ripple activity in the hippocampus, a lot of that looked very similar, really, to what we see during REM sleep. We see a lot of this cortical activation during REM sleep, and so in order to possibly, at least, in order to block REM sleep, the locus coeruleus and raphe nuclei are being suppressed in some way during these ripple events in the hippocampus. The take-home message from all of this is basically that these ripple events during slow wave sleep seem clearly related to some sort of memory consolidation effect. We saw that there's a strong correlation between the number of ripple events and the amount of learning that participants are able to show. And in addition to that, then, and that was shown by Lewis in the schema talk that I talked about. And then in addition to that, Logothetis managed to show that these ripple events were seem to lead to preferential activity and preferential connectivity between the hippocampus and the cortex, suggesting that during these ripple events, the hippocampus is actually kind of transferring information to the cortex, and we're actually able to, and Logothetis is actually able to see that happening see that happening live with his methodology of neural event triggered fMRI, by just waiting for these ripple events to happen in the hippocampus and then recording what is the fMRI activation immediately after these ripple events. And so I just thought that was really a really nice study, just able to almost see a memory being consolidated live, and that's pretty exciting. All right, so that was what happened on day 4, and so day 5 now, and check back in later, and that's it for right now.